Regulating the steps of development is done through the use of pathways. There are two major types of signaling pathways, the paracrine signaling pathway and the juxtacrine signaling pathway. In juxtacrine signaling pathways, receptors directly interact with receptors in other cells to induce changes in the cell, including changing the state of the cell. Paracrine signaling pathways involve a ligand binding to a protein involved in cell signaling. This results in a signal transduction cascade which causes changes in the cell. There are four major types of paracrine signaling pathways: the fibroblast growth factor, or FGF, the hedgehog pathway, the Wnt pathway, and the TGF-beta pathway, all of which are important in development. We'll be focusing on the hedgehog pathway. In the hedgehog pathway, there are three members: sonic hedgehog, desert hedgehog, and Indian hedgehog. Desert hedgehog is important in the regulation of spermatogenesis, and Indian hedgehog is important in the regulation of cartilage growth. The role of sonic hedgehog is more pervasive. It works with FGS to regulate many different processes. One example of a role of SHH is in its expression in the developing nervous system. Too little sonic hedgehog can result in cyclopia, or having only one eye, while too much sonic hedgehog can result in cancer. The sonic hedgehog pathway is activated when hedgehog binds to the receptor patch. Normally, patch binds to smoothin, inhibiting smoothin. Cubitus is tethered to the microtubules of the target cell by COS2, and with the help of pKa and S-lim, it's cleaved in such a way that Ci enters the nucleus and prevents transcription of the hedgehog responsive gene, suppressing transcription. When hedgehog binds to a receptor patch, it changes in shape. Patch can no longer inhibit smoothin. Smoothin is now free and can bind to pKa and S-lim, which frees cubitus interruptus. The function of the Ci then changes from a repressor to an activator. The cubitus moves to the nucleus, where it binds to another molecule called CBP, and it activates transcription. To sum that up, when hedgehog is introduced to the pathway, it represses patch. This in turn frees smoothin, which is normally repressed by patch. This inhibits pKa and S-lim, which under normal circumstances are repressed by smoothin. These normally inhibit the cubitus interruptus from being present in its entire form because it cleaves it, which represses transcription. The cubitus interruptus is then freed and can proceed to activate transcription of hedgehog responsive genes. Sonic hedgehog is important in regulating many steps in development and is shown to be involved in the formation of cancers. Our knowledge of the sonic hedgehog pathway can help us prevent many problems from occurring, and learning about it is important in the understanding of developmental biology.